I'd like to first thank the organizers for giving me the opportunity to tell you about my work. So as you will notice, the title of my talk is changed from what is on the program. So I apologize for that. Towards the end of my talk, I will talk a little bit about collective cell migration on curved surfaces. But the story I'd like to tell you is another piece of recent work which looks at how mechanical heterogeneity in dense tissues alter the overall rigidity and the properties of cellular invasion. So to start, I'd like to just show a few examples that probably most of you are quite familiar with of collective cell migration. And so here are examples of wound healing, which we have seen a little bit also early in the morning, and developmental collective cell migration, as well as tumor invasion. So one common theme I'd like to just highlight here is that this cell migration is happening while the cells are remaining densely packed, meaning that they're not moving individually to form different structures and perform the functions. They are doing so while essentially holding arms with other cells and moving as a collective or a group or a cluster of cells. So what I have been doing in the past several years is looking at what sort of physical properties you can expect at the tissue or global level during these situations. So if you think about it, for example, epithelial cells that line our surfaces and our skin and organs, essentially during most of the time behave pretty much as a solid type of material, meaning that they're supposed to provide a very tight barrier function, prevent things from getting through, and they're really not moving around in these situations that you see. But under perhaps disease conditions or under developmental and even tumor development, these can actually alter their collective state, meaning that there's some triggering into a kind of a fluid-like response where these are now moving and the relative position and packing of these cells is no longer preserved. So this kind of apparent switch between these different states is quite interesting, even from a sort of a soft matter physics perspective. And indeed, some of what already has been done in thinking of densely packed objects from soft matter physics has been applied to think about these type of transitions between quiescent epithelial and sort of collective migratory states. So here's one example, and this is from work that's a couple of decades ago, looking at the collective behavior of attractive colloidal particles. And of course, these are totally not biological. And in short, what this is essentially a phase diagram is a function of the packing density of these objects. So remember, in all of those biological examples, cells are densely packed in essentially a confluent limit without any gaps between them. So what happens to a non-biological material as you change the density? Can we understand something along that axis? And here, there's also temperature and applied deformation. So those are also axes that control the behavior of these type of densely packed materials. So what this surface is essentially is a boundary between what you might expect to behave as a solid versus outside of the boundary where it's behaving more like a fluid. So by changing any three of these parameters or a combination of them, you can transition between a fluid and solid-like behavior. And indeed, this kind of framework has been applied to looking at cell movements and the material states in biology. And this is one of the recent works from the group of Auger Campos in looking at in vivo, in the zebrafish, whether cells are behaving solid-like or fluid-like. And one thing I'd like to point out is that they find that in different parts of the embryo, you can essentially map the behavior to one that is sort of inside that phase boundary, so behaving like a solid, and another data point which is outside of that by changing the combination of volume fraction, active fluctuations, and supracellular stresses. And this kind of, if we focus on this axis over here, which is the packing density, you can see that if you increase the packing density, there is a critical sort of a packing density beyond which you become a solid-like state. And this has also been explored quite a bit in recent years in, for example, in a monolayer of MDCK cells, where essentially this is proliferating over a period of about a few days, and they go from that initial fluid-like state where cells are rearranging and collectively moving to a final so-called jam state. And that word has been used a little bit more and more to describe what is happening in this kind of that final state. So these cells are getting into a jam, essentially coming from the word traffic jam, where cells are caged very much by their neighbors, and they're unable to basically overcome the resistance that comes from their neighbors. So that final state behaves very much as a solid. So these are just some of the examples that show this kind of a density-driven jamming transition. And recently, we've also looked at a couple of examples where this kind of transition doesn't necessarily just happen as a function of changing density. In fact, this can happen even without a change in the overall packing density. So this is cells that are cultured from cells that are taken from human patients, and these are the human bronchial epithelial cells that typically in vivo would line the airway in the lung. So on about day 0, these cells essentially form a confluent sheet. So what we're looking at is way after it becomes confluent on day 3, and you see that the cells are moving, sort of sloshing around, and there's also localized rearrangement going on. But on day 6, the number of cells, sort of the packing density here, is remaining pretty much constant compared to day 3, but the activity has slowed down quite a bit. And finally, on day 10, you can see pretty much all the activity has ceased, and if you look at the position of each cell, it's essentially stuck in place. So this is a kind of a jamming that is happening without a change in the overall density. And just to show that this state is not some kind of singular type of behavior, you can in fact take that state and apply different perturbations to it. And in fact, you can reverse it back to a more fluidized state. So in particular, this kind of perturbation is simply apply a increased pressure in the air-liquid interface that is used to culture these cells. So after applying a slightly heightened pressure, you can trigger this kind of fluidization again in this tissue. So I'm not going to go too much into this, but except to show that these are just some of the examples where fluid and solid behavior can exist due to various factors. And in fact, recently, we have some work on the bioarchive, which shows that the kind of traditional way of fluidization for epithelial cells, which is the epithelial to mesenchymal transition, is a distinct path compared to what I just showed you, which is that you can trigger a kind of fluidization by simply an application of a increased pressure in the culturing chamber. So these are quite distinct, and they're interesting signatures that you can pick up to distinguish it not just from a molecular level, but you can also distinguish the two processes from a morphological and mechanical level. So like the title of the beginning of the talk, I'd like to tell you about yet another path in which you can control the overall rigidity or fluidization or solidification of a dense tissue. So this is where I like to just briefly highlight the intra-tumor heterogeneity in cancer. And as you all know, cancer tumors are notoriously more heterogeneous in many ways. So typically, there's a lot of focus to explore the genotypic heterogeneity, even within the same tumor. And there's more and more work which explore the phenotypic heterogeneity. And what I like to focus on is sort of the mechanical side of this hallmark of cancer, which is the mechanical phenotypic heterogeneity. So there are recently quite a few experiments that try to explore precisely just how mechanically heterogeneous are cells within a given cancer tumor. And there are different types of methods developed. So this is sort of a more traditional method. You just kind of take an AFM probe and take a cancer tumor and probe at various spatial points. And from that, you can essentially get a mechanical phenotype of the individual cells. So in this case, I'm just plotting a histogram of the stiffnesses. And you see that in cancer, you get a wider distribution of stiffnesses compared to the healthy counterpart. So there's another type of method which is used by one of my collaborators, the group of Joseph Kess at the University of Leipzig, which is essentially developing a microfluidic device which is able to, under a high throughput method, squeeze or perturb every cell that comes through. So you can take tumor cells and flow it through this device, and on every cell you can exert a certain amount of force. And then you can measure how much deformation there was for each of those cells. So if you measure the relative deformation, basically defined as the ratio of the stretched shape compared to the unstretched shape, you can plot a histogram of the relative deformation. And you can see either whether you're looking at breast cancer or cervix cancer, overall the cancer cells are softer. That's something we sort of known all along. But another interesting thing is that the cancer samples are usually more heterogeneous. So there's a wider distribution of these deformabilities. And that is even more pronounced in the cervix case. OK, so this is really showing that perhaps this is almost a universal feature of many cancers. And from a modeling point of view, how do you begin to think about the mechanical heterogeneity? So most models usually model cells using agent-based models, typically don't take into account this kind of inherent mechanical heterogeneity. And this is what I like to do in introducing a very simple, quick model to think about this kind of heterogeneity. So you may have seen this class of models, which are called Vertex-based models, used to describe a 2D monolayer of cells, or it can be generalized to a 3D packing of cells, let's say in a tumor, where essentially each cell is described by their geometric shape. And in 2D, it's really easy to describe. So if I'm looking at a 2D monolayer, then these cells form a polygonal tiling, pretty much as like tiles on the floor. And the vertices where the junctions meet are the degrees of freedom in this computational model. So the goal is to essentially to describe the cell shapes that you see and overall the mechanical response by first prescribing this model with a mechanical energy function. So here, let me just introduce you a few of the terms that we put into the model, and I should say the model has been around for quite some time, originally from a couple of decades ago. So the idea is that you take a look at the area of the cell, and essentially in a monolayer, the 2D area is not allowed to fluctuate very much. And that is due to actually the 3D volume of the cell. Think of these cells really as packed columns of 3D cell shapes that are confined to a constant volume. So if you assume that the volume is conserved and then the height is constrained to fluctuate, then that will result in a quadratic term in the area. So this means that the area times the height of the cell gives you a constant volume. And if the height is coupled to other cells' height quadratically, then that can also be written as a quadratic area term. So another ingredient is the cortical tension, the contractility of these cells, and this typically comes from the existence of actin-myosin rings at the periphery of the cell. And in the lowest order, you can just give it an elastic nature, which essentially says that the perimeter of the cell is acting very much like an elastic ring, which controls the shape of the cell. And lastly, there's a very important term, which is linear in the perimeter of the cell. And you can think of that, energy linear in the length, you can think of that as a line tension type of energy. And that is modulated by different effects. And the important two is the cortical tension itself, which is trying to basically minimize the perimeter of the cell. But there's a competing term, which says when a cell is in contact, when it forms a junction with another cell, that the adhesion molecules on the junction will tend to lower the effective surface tension or the effective cortical tension. So overall, the competition of these two results in an effective line tension on the perimeter of the cell, which we call gamma. So all of the work that was done before basically considered that all of these parameters to be homogeneous, meaning that every cell has the same set of parameters, the same set of preferred area, the same set of elasticity of the cortex, as well as tension and adhesion properties. So this was an early paper by the group of Frank Ulischer, which looks at basically what type of ground states do you see within this model. And a little bit later on, I also worked on describing sort of the amorphous states of the model and describing overall the mechanical behavior. So we basically discovered that as a function of this preferred P0, which comes from this gamma here. So if you assume all of the cells have the same set of properties, then you can non-dimensionalize that energy function, and that results in a preferred perimeter P0 for every cell. So by changing that P0, we discovered that there is a very clear second-order phase transition in the rigidity of these tissues. So that was able to offer an explanation for this kind of a density-independent transition from a rigid solid-like state to fluid-like state. And recently, together with Le Yan, who's in the audience here, he's now at the BioHub, we came up with the theoretical framework to really explain why you should expect a rigidity transition at all. So, but basically, the point of this is to say that with this kind of model, it's very easy to get a kind of transition between rigid and fluid-like state. But basically, this kind of heterogeneity that you would expect from a cancer tumor is not typically included in these type of models. And usually, if you, before I did this, even I thought that probably just adds another form of noise and probably does not affect the results too much. But then when I saw those sort of experimental histograms of cell deformability, they're really much wider than typically considered. So what if you actually assume that level of heterogeneity from a cancer tumor? And what happens when you introduce that kind of heterogeneity into this model? So the way we introduce it is to say that every cell which has a preferred perimeter coming from the mechanical tension of the cortex and adhesion, we're going to assign to it a different value of P0. And that distribution of P0 is taken just from a very, the simplest you can write down, just a Gaussian distribution of these parameters. And what that essentially results in is that the mechanical response at every cell can be quite different. It's essentially picked out randomly from this distribution with a mean mu and a standard deviation sigma. So here's just that's it? Okay. So, all right, maybe I'm sorry, when did we start? We started much later than 3:30.
I'm sorry, that was the 20 minutes, and then you have 5 minutes of Q&A.
Oh, okay.
Yeah.
Okay. So, so then I still have to. So the point is that in this model, that when you add heterogeneity, you essentially. So here's the baseline. Here's that rigidity transition I described as a function of mu. And that happens when sigma is equal to 0. So no variation, no heterogeneity. And by introducing heterogeneity into this model, we find essentially two things. One thing is that it always seems to rigidify the tissue overall. That essentially says if you increase sigma, you can always increase the overall shear modulus of the tissue, meaning that it rigidifies. And I'll maybe skip ahead. Okay. Maybe this is a good point to summarize. And essentially, what we find is that in the presence of heterogeneity, we essentially discover that there is two types of processes which are controlling the overall mechanics. One is the rigidity transition as a function of rigid cells, and this can happen as low as 20%. This means that the amount of rigid cells that you need to get a rigid tissue is only at 20%, but this is happening even before the onset of the contact percolation transition. Okay, so I think this is, okay, so I think I'll just maybe end with this. So this is what I originally planned to talk about, but I think we're not quite ready to talk about this yet. But I do want to show that it's really also interesting to think about the collective cell behavior in curved space. And this is one example where things are happening on a spherical surface. So this is a monolayer of cells that are embedded to a curved sphere. And there's, of course, many biological motivations for this. And one thing we're really interested in is what brings you the onset of collective coherent rotation around the sphere. And this is even happening without any explicit alignment between the polarity of the cells. So this is something that we are very excited about, but hopefully we'll be able to report the results soon. So with that, thank you for your attention.
Does the shape of the cell matters in your Vertex model when you have these edges with the Gaussian distributed force?
Yes, so the shape matter. Can you?
You're using the hexagon, for example, using different shapes, heterogeneity shapes of different cell shapes. Will that change the result? How does it change the result? Like if in your simulation.
So the shapes are they come out of the model, right? So the model simply assumes how cells interact with the mechanical energy, and then shapes are measured as a result of that.
Oh, sorry, I didn't understand.
I think we have time for one more.
I'm wondering, are there any non-biological models which show a solid to liquid transition as you increase in homogeneity?
Not that I know of, but I may not have heard
Yeah.
published
Okay.
results.
I think we can do one more. We're just going to keep doing this until we've actually run out of time.
Going back to the first question, is it the shape that matters, or is it the three connectivity of the vertex? Because you had that funny percolation transition at 50%, which is a result of triangulated models if you do the Voronoi type.
So this is actually always assuming that only triple junctions are stable. So if you look at these snapshots, there are no fourfold junctions, no T1 junctions or rosettes. which we have only explored also recently. But this is assuming only triple junctions are stable. And then that basically gives you this two different percolation processes. Yeah. So I think this is true for most confluent tissues, except for special situations where you have fourfold and higher.
Great. All right. Let's thank Max one more time.